Hi, everybody, and welcome. Thank you so much for coming to our Data Science Institute Distinguished Lecture by Baroness Ingrid Daubechies. I wanted to mention that this is one of a series of lectures that we're hosting, and some of the other speakers and the dates of their talks are listed here, so please do come back for some of our upcoming lectures as well. As I'm sure most of you know, Ingrid is very well known for her fundamental contributions to many areas of mathematics, maybe perhaps most famously with wavelets, as well as her interdisciplinary collaborations ranging from art to biology, and we'll hear about some of those today. She's been highly decorated and recognized for her contributions, including earning the National Medal of Science. I just wanted to say a couple of things, actually, that you won't find on Ingrid's Wikipedia page. I actually first came to know of her when I was an undergraduate interviewing as a prospective graduate student at Rice University. And Richard Baranuk showed me a double pendulum that was an example of a chaotic system. And he was playing with it with me when I was interviewing, and he said, oh yeah, this was a gift from Ingrid Daubechies, and it's just fascinating, and it's really helpful for me to kind of visualize and think about different interesting mathematical problems. And then I remember years later when we were both at Duke University, we were at a joint conference and our flight got canceled, and we decided to drive overnight back to Duke because Ingrid really wanted to be able to teach her class the next morning. And I was just really thrilled to get to know Ingrid and about all the different scientists she'd worked with and some of the just guiding principles she'd used to think about what problems and what collaborations she valued. And I think that's just evident through her whole career. Everyone who's had the privilege to work with her has benefited from knowing her. So I'm just really delighted to have you here, Ingrid. Please help me welcome her.
Well, thank you, Beth. And on that memorable drive overnight to Durham, Becca was also a sterling navigator because her phone was almost out of juice and we had to keep it going until we were home. It worked perfect. Okay, so this is a talk that that documents an effort that has been ongoing for many years, where I work with biologists on biological data using diffusion methods and see. So because I didn't know the extent to which people in the audience would be familiar with manifold learning, and so I have a little intro about that. So a manifold may have a much smaller dimension than how it's first presented to you. So here you have this cartoon image of a two-dimensional thing in 3D, as the shadows show, and you just get a 3D picture. How would you know that it's two-dimensional? Well, I really think of the method that I'm going to propose, as similar to having all these little sequins on these animals. Each one of them is completely flat, and together they give you an idea of the whole animal. So we're going to do the same thing. We're going to, on our blue thing, we have data, and we're going to take neighboring data, and because we assumed manifold, I'm going to not think of singularities in this talk, because we assume it's nice and flat. Neighboring things will approximate the tangent plane. So nice and smooth. Neighboring things will approximate the tangent, a little tangent disk like the sequin. And we'll have many that overlap. And together, they will help us follow the whole surface, like the sequins on the animal. So how would we do that in practice? In our data set, if we take points, then for the same reason as before, when they're very close to each other, I do trust those distances that I measure in my ambient space as being truly reflective of distance on a manifold, but distances over large things that are further away, I don't. Now, I know you can immediately think of things that make ruffles and they're close and so on, so there will be exceptions. just think of the general principles so far. Small distances are generally trustworthy, large distances are not. But what I can do is I can use the fact that most small distances are reliable in order to explore this data set. And what I do is I stay in a little neighborhood, then I can go to another neighborhood, and so jump to this other description, and then look at this, and so on. And so that way I can travel in a trustworthy way over the collection and travel large distances. So in the first approximation, because I have this underlying unknown manifold, what I'm going to exploit is that if, as in this drawing, if I have 2 points on my manifold, then either the chord distance, so the distance in the ambient space, or the geodesic distance measured on the manifold, or in the tangent plane on one of the points, the pullback of the other points, and so on, and the distance to that, in first approximation, these are all equal if I look at small enough. So what I'm going to do is I'm going to write a heat diffusion kernel using the distance that I can measure, take it as an approximation for distances that I don't know on the manifold, and I'm going to just use that distance to build a heat kernel. Just the way I would standardly do. Implicitly here, I am also exploiting that the heat kernel has the same expression in Euclidean spaces, regardless of the dimension. So I write the expression that I would get in the ambient space, but that's also the expression you would have on that pullback of the tangent plane in first approximation. Now, of course, the normalization depends very much on the dimension, but the normalization, for the moment, I'm not caring about because it will have to do a lot with the sampling density and so on. So I'll come back to that in a moment. So the size of T, so that measures the extent of the diffusion. So I take T typically very small, and I'll come back to that as well. And then what you can do is, if you want to diffuse from one neighborhood to another and so on, you can just look at that small diffusion and apply it many times, which means that you look at powers of this matrix. So, which means that the spectral representation of this matrix for small t will be important to understanding powers. Okay, normalization. So, what we want is something we'll work numerically. We want to define a random walk on our data. So we want something that should be something that takes the vector of all ones to the vector of all ones for normalization. And that will take into account the fact that my normalization should be adapted to the dimension of my manifold and to sampling density that may be different in some points than others. I compensate for that. So I just take my matrix in such a way that every row adds to 1. And so this is how I build my matrix. There are other instances, and in fact, which do a little better if you don't do a one-sided normalizations, but if you try to make it a double stochastic matrix. But that's a later refinement. Then there's also, so we've taken care of normalization, and we've in one fell swoop done for dimension and for sampling density. But how should I pick T? I should pick T not too large and not too small. Not too large, because since the exponent is the distance squared over T, if T is very large, then even very large distances will still give me appreciable matrix elements, and I don't want to take those large distances. So T should not be too large. But T can also not be too small, because I have my data sampled. And if I take T very small, if I had it really smoothly, then it's true that a small T would diffuse everything a little bit. But if my T is so small that I never feel my neighbors, well, then nothing happens. So I need to take T large enough, and not just to feel one neighbor. Actually, I need to knit together all my data in some sense. So in this method, many people have told me that they felt choosing the right T was more an art than a science. And one of my graduate students, actually, in her thesis, developed a method that works very well for us. And the idea is that you want this thing to behave like a diffusion. Well, what a diffusion has as a property is that it's a semigroup. So what you do is you build your approximation to this diffusion, this numerical diffusion, and you test how different the diffusion squared for t is from W to t. If it is really a nice semigroup, then they should be close. So in practice, what you do is you look at the difference of the norm of this matrix. You try to get away with t as large as you can so that this difference is small. So she tested that out on the data. This is just a well-known test data set of a helicoid on a helix on a torus. And depending on how you choose t, the embeddings in 2D that you get from this may look like these three examples. I mean, that's why people said it's more of an art than a science, because you can look at things that are really terrible. And for instance, in the rightmost one, embedding, it's clear that you took distances that were too large into consideration because you kept that going closer and wider in your picture. But if you look at this way of, like looking at the deviation from semigroup behavior, then you see that the right one is the one that is favored by that difference. Similarly, this example, Swiss roll, again, you can get these different kind of pictures of what the 2D truth looks like. But again, if you look at that semigroup behavior, it singles out the one that is closest to the rectangle that you know is the ground truth. And I've known, since she published this, Shan Chan, who is now a professor at the University of Southern Denmark, this has been taken over as a criterion by many other groups who find it useful for them as well. So, we have taken care of how to pick t, and how to normalization and pick t. And then what we're going to do is we're going to do exactly what I said earlier. We're going to take this matrix and we're going to look at its spectral decomposition. In fact, here on this slide I made a slight mistake. The WTIJ there is a symmetric decomposition, while the matrix WT is not a symmetric way of making it a random walk. But you can make it a symmetric walk by multiplying right and left, a symmetric matrix by multiplying right and left by d to the t to a half, and then you have a symmetric. You then have something that is symmetric, and you can you have a similarity to a symmetric matrix, and you can then from the symmetric matrix do the transposition, and you get the spectral form. In any case, yes.
Quick, just a clarification question. So, just trying to understand what you're trying to do. Usually, when people do diffusion, they try to make the process have some generalizability that doesn't land on exactly the data points that you know with them. So, you're building a random walk on the data points.
Yes.
So, what is the motivation behind that? What are you trying to get at?
What I'm trying to get is a way so I'm wanting to get this spectral decomposition, which will enable me to give a different parameterization of my manifold. So that's what I want to get to. What I'm saying is the manifolds I will have are noisy, and a whole lot of the structure is hiding in there. I hope that by building in something that takes the structure into account, I will denoise. And so here, by building a diffusion on the data points, so I'm not trying to interpolate between data points. I want to find the structure of the whole thing so I can denoise.
Okay, thank you. Yeah, that's very good.
And that's what I'm doing. So I take, and I then can look at powers of this diffusion operation on the data points by looking at just decomposition. I typically truncate after a certain number of eigenvectors that I estimate depending on the data, and I take powers of the eigenvalues. And then that gives you a parameterization for the data points. I mean, every data point, I just take the appropriately weighted eigenvalue and the value of the eigenvalue of an eigenvector in that data point. That collection of vectors is the collection of parameters that I would give to the data point. So I make an embedding in our capital L, where L would be the cutoff of my. And capital L is typically larger than the true dimension. I'm not trying here to actually estimate the dimension. I'm trying to get a parameterization that, because it took into account to some extent, the manifold structure, will have denoised everything not consistent with my way of parameterizing. And you can then, in that space, look at a diffusion distance. And that's a method that was first proposed by Ronald Koifman and Lafont in Yale, and exploited by Mauro Maggioni also extensively. And so it's also represented in papers by Belkin and Yogi earlier and independently. And what I'm going to show you is application of this idea and an extension of this idea, which I'm very enthusiastic about, to a set of biological shapes. But let me now backtrack a little bit and tell you about the biological shapes. So it all started with a conversation with a biologist, with Jukka Jörnvall, who is a distinguished Finnish biological morphologist, a primatologist, and Daubechies, who was then his graduate student while he was visiting his postdoc, while he was visiting Stony Brook, and who's now completely, by coincidentally, a professor at Duke. So it really has helped that we ended up at the same institution. And the first conversation was that they told us they studied the teeth and bones of extant and extinct animals. And Jukka Jörnvall told me about some work they had done that had caused a big splash on using a characterization of the complexity of the chewing surface of molar teeth in mammals. So what he told me was that they had found a way of characterizing the complexity of those molars, of the chewing surface of the molars, that made it possible to cluster together to distinguish not just between herbivores and carnivores, that's easy, but between people among the herbivores who ate plants or flowers or fruits, and in the carnivores between animals that ate insects or that ate red meat. So he was and it was also a characterization that was independent of the size of the animal. It worked for shrews, it worked for elephants. So this had made a big splash, it had made a paper in Nature and so on. And so he was telling me this was a social occasion. So because he heard I was a mathematician, he said complexity of surfaces. He said, how do you measure the complexity of surfaces? And we're talking early aughts. So he said, well, I had seen on somebody who was doing computer graphics representatives of surfaces, and at that time we had these sun systems, you remember. I mean, some of you may remember. The ones with gray hairs among you may remember. And where a surface was rendered by having virtually a red and a blue and a white lamp, and you were seeing the shades of those reflected colors on the surface, and that led your eye to integrate what was going on. They said, oh my God, colors give you and so on. And so what they then did was they took their T surfaces, looked at them in this rendering from the top, took a screenshot, they had fine triangulations, they blew it up, and they started counting the color transitions between those triangles. Those different triangles, they were all flat. And I said, oh. And they had a name for what they were doing. But what you're doing really is looking at the normal on each surface and how much its angle changes from one triangle to the other. He says, I guess. And I said, well, mathematicians do this and have computational methods for computing things like that without having to do this color computation. So he said, oh, that would be great, because that would be. And it's not that they're counting by hand, I mean, but still, was much more. So I developed with collaborators, and here Jaron Lippmann at the bottom at left was then my postdoc. He's now a big deal professor in the Weizmann Institute, and he was just coming to me as a postdoc. And computer graphics then and now is a field that's very much fueled by the large amounts of money in then only animation and now also computer games. And so it's a field where people develop algorithms and so on, but they're all aimed at those applications. And he was thrilled to be working on something that would have a scientific application. So he was enthusiastic about it. He wrote a software that did exactly that. I mean, take the surface, compute, integrate over the surface, change in angle of the normal. They called it Dirichlet normal energy, DNE, because DNE stands nice to biologists. And it became a paper in Nature Methods. And, but so that was the beginning. Here I'm giving you a list of people who've worked with me over the time. Let me just say what their fields were. Rima Ifari came from mathematical methods for inverse problems. Doug Boyer, we've seen, he's a morphologist. Tingwan Gao was a fantastic graduate student I had at Duke who spent a few years in Chicago and now works, unfortunately for him probably, in finance. Roy Coran was a postdoc who was a student of Jaron, who then went to Zurich and is now back in Israel. Jesus Puente, another graduate student who ended up in finance. Robert Ravir works for, and here further, Shan is the one who came up with the beautiful method for finding T based on the semicolon property. And so we have here computer scientists, mathematicians, and one student who, as an undergrad, worked with us and then is now in grad school in neuroscience. I like to show them in the middle or at the beginning because usually I run out of time and I don't want to fold them off the presentation. So that first project was a complexity of teeth, and it was not really an off-the-shelf thing, but it was very much, mathematically it was an off-the-shelf idea, if you want, in order to do computation. But then, as often happens, then the interesting questions come. So the next question was, wow, if you can do this, this was wonderful, this is great, and they're still using it. Can you help us with this other thing? And that's actually something we have been working on in a different form ever since for the last 10 years. So the data they use are data, they actually do micro-CT for their data. And you might think, well, CT is something that gives you volume information. And we're talking about surfaces. Why don't we use the whole volume data then? Well, most of the CT they do is not on the teeth themselves. Doug Boyer is very interested in all kinds of mammalian teeth, and especially primate teeth. And whenever he goes anywhere in the world, whether it's for vacation or a conference and so on, he looks up all the local musea in our collections in the neighborhood, and he goes and visits them in order to get all their primate. Many of the most interesting teeth are things that are very rare, and they're not going to lend. It's theirs. But they will let an expert take a cast. And so he uses very fine dental resins to make casts of these. And he comes back with all these casts. So in what he has brings back, the surface is the only true information. All the rest is nothing. So that's why they do CT, then they extract the surface, and they reconstruct. And then we may sometimes put a texture on it to make comparisons. So the way they typically work, and most morphologies, many morphologists still work, is to, if they want to study something, is they take the collection of things over which they want to study whatever they want to study, and they look at what this collection has in common, what points are homologous between all of them, and they landmark them by hand. Well, now on their computer, but still by hand. So they put in points that they identify, and they then use the spatial coordinates of these landmarks as features. So they build a feature vector of 3K entries, which are the spatial coordinates of these, and that represents the shape, and now they use that vector as a proxy for the shape, And here is a collection of teeth that have been landmarked by experts like that. And what they will do to define a distance between two teeth is then move one as close as possible to the other one, so that the R3K, and then they look at the in R3K at the distance vector of these two R3K tuples. So the landmark placement itself is something that's tedious and time consuming. So in general, they actually don't, they have, Doug has many more teeth in his collection than he typically uses, even to illustrate a certain feature, than he uses to write a paper on that feature, because it's so annoying that they do it just until they have, if their hunch was true and they can indeed prove a hypothesis, they do it until the p value is below or above or whatever the value that the journal will accept, and that's it. I mean, and all these other data. So if there were a way of doing that automatically, it would be so much stronger, because you would use all those data. There's a lack of flexibility because you need the same number of landmarks on each example. And what that means is that if you have one of those is a very precious fossil that's chapped. That means that on that piece, the chapping piece, the piece that's chapped there, you cannot put any landmarks on the other teeth, even though it would be useful in their comparison, because you need a high degree of expertise, and so it is not easily accessible. One thing that Doug has done is he's created something called morphosource, in which people can upload digital data of surfaces, and people are now doing it even for archaeological data, so things that are not in this field, and use tools that are available to compare them. But you can't do that automatically if you have to first know a lot about what you're looking at. You can't say, oh, I know everything about the femur, let me see what happens in the teeth of this animal, if you're not an expert on those teeth. And even debates exist even among experts. So they asked us, for all those reasons, can you find a distance among things that we have already studied mathematically that would do well for us? And so the way they define their distance is, as I said, you do a rigid transformation on one data set, and you then sum after minimizing. And they wanted to find a distance that does as well as their Procrustes distance does. And does as well biologically. I said that here because the first thing when I said, mathematicians have zillions of distances between surfaces. This is well studied. There's Gromov has, and so on. I said, yes, I said, and for every one for which software exists to compute it, we've tried it, and it just doesn't do well for our data. We can get more out of our Procrustes distances, which of course have input from us domain knowledge, than out of those purely geometric things. So we would like a distance that does well biologically, that does at least as well as what we do with our landmarks. And can you do that? And so that's what we did. What we first, so I'm going to give you a quick show of two distances. But what we realized, well, let me just show these and then come back to that. So we defined actually two different distances. One which I liked mathematically better than the other, except that the other then did well better in practice. So we live with reality. So in the first one, we defined what we call the conformal Wasserstein neighborhood distance. Okay, so we first flattened all these surfaces conformally to a disk. The reason for that was not because we believe that conformal mapping is so important for biology, even though that has been asserted by mathematicians. It's just because it made it possible to do search in two-dimensional space instead of 3 dimensions, and on a common domain. The conformal flattening gives you a landscape on the, which perhaps in the conformal flattening you preserve all angles between tangent vectors, become angles in the plane, and then what happens is that in doing that you have to shrink or expand, and so we keep track of that factor. That includes, that characterizes, that encodes a lot of the geometry. So that conformal factor we viewed as a landscape on the disks. And we then, if you want to do a Wasserstein distance, you have to look at how similar or how dissimilar is one point to another on the two. And so what we did is for each of those points, we looked at a neighborhood in hyperbolic, and they really should be circles, non-centered circles, but circles instead of these potatoes. But now the thing is, conformal flattening is not unique. Once you have it on the disk, any Möbius transform will transform the whole thing to an equally good conformal picture of the original surface, because Möbius transformations on the disk are conformally invariant. So what happens is that we exploited that we needed to find a distance that was independent of choose what conformal thing we do. But we exploited that by saying, well, conformally we can bring on each of these two disks, we can bring the 2 points that we are trying to compare to the center. And then the neighborhood just becomes a little disk around that point, the center. And so what we do is we compare the landscape that we have conformally brought to the center between the two. You only have to take a rotation. That's just easy. So that distance of similarity or dissimilarity is easy to define, and it's now invariant of what conformal flattening you took. Then you can use that either here like an L1, or you can use L2. I mean, you can use that in a Wasserstein approach to find a distance, and we defined that the conformal Wasserstein neighborhood distance. And I loved it, and we could prove nice theorems about it and so on. But it was computationally kind of heavy. And so we also tried to go back at what their progressive distance and said, look, what does and see whether we could find an analog, computational analog of that. That what does their distance really do? It says if you have a correspondence between the 2 points, then what it does is it compares the for a given correspondence, it compares the corresponding point to where you move by rigid transportation, your point on the first manifold, and look at the distance between those. So you take the 2 green points, if you wanted, when you had landmarks, and you move them, and you look at their distance squared. And you integrate over the whole surface. That's if you have a correspondence. Well, what correspondence are you going to look at? Well, you want certainly something that's smooth. And we decided mathematically to define it as area preserving. And the biologist says, well, area preserving, we don't believe in area preserving. Of course they don't. I said, but how in practice are we going to implement this? Are you agreed that we have to put points all over the surface in one and all over the surface in the other? I said, yes. And we have to distribute them reasonably. They said, yes. I said, well, one way of seeing that they're reasonably distributed is for each point to look at what zone of the whole surface is closer to it than to all others. So it's a little Voronoi set. You're going to put equal points on each, 500 on each. And we also normalize because we wanted to compare elephants and shrews, remember? So all the surfaces, all the teeth are always normalized to 1. So we have surfaces of area 1, both equal numbers of points, and we want a correspondence where we map really a little Voronoi cell of 1/500 to another one of 1/500. So in practical, I mean, you may not want to call it area preserving, but it's going to be area preserving. I mean, it's an approximation to area preserving. If we define it that way, then we have a mathematical object that we can deal with, a smooth area preserving map, and we can prove theorems about it. In fact, what we could prove, which was very nice for our computations, was that if you define, and we then minimize over all the C's, of course. That's illustrated here in this curve. Suppose that I have this correspondence for a blue and a curve and a red ellipse. Then reorienting the curve, so applying a ridge transformation, gives me a smaller map, a smaller distance. But that's not the best way of doing a correspondence. The correspondence on the right actually gives me a much smaller distance yet. So I have to minimize over R, which is easy, just an SVD, and then minimize over C. Minimizing over C in practice is horrible. I mean, the area preserving maps are a truly awful set of things to compute with. However, we could prove that if the distance is smaller, remember, small distances are more trustworthy, then the minimizing area preserving is very close to conformal. It's actually very close to the minimal conformal map. And so that's what we did in practice. We looked for the closest conformal mapping for things that were, well, we looked for follow, but we only trusted the outcome when things were close at the end. And so we were close. Okay, so we defined our distances. And our distances come, if we do continuous progressive distance, with a mapping. We use these two distances. In the paper that got published at PNAS, we had them, biologists are very insistent, rightly so, that prior to doing anything, when you define what success will look like. I mean, there are times that you can cherry pick and look and so on, but that helps you to define a hypothesis, and then you test the hypothesis with a preliminary definition of success. So one definition of success that we had defined was that we would put our distances in a matrix where we would compare the observer-determined landmark placement distances with the distances we computed. Blue is very small, white is medium, and red is large. And we ordered them. This is many more teeth and bones than only lemur teeth, which was originally the goal. So the matrix is just teeth, mammalian teeth, and we numbered them so that things that biologists felt were similar were labels that were closer to each other. So that means that we wanted the structure close to the diagonal to look the same as theirs. And indeed, you see that it looks pretty good for both, but better for the continuous progressive distance than for the other one. So from then on, we worked with continuous progressive, which was also the easiest. They also wanted to see if you took landmarks on one and we transported with our map to the other, whether that corresponded to locations of the that would be observer placed in the other one, and it did pretty well there. So we had and we published it in a nice journal. Okay, so yes.
So the two distances, are they? What's the complexity? Are they n cube or square? Do you remember? No, that's okay.
The one for CP distance, I think, is n squared.
N squared.
But the computational Westerstein neighborhood was at least n cubed.
Okay.
So, but I mean, that's what I remember. But, okay, so and Ting Ram then said, so several things happened actually. First of all, we had to produce these mappings as a byproduct of finding good distances. So the distances we got was the distances at the back, at the left here, the continuous progressive distance. And I should tell you what you're looking at. You're looking at a collection of 50 primates in 5 genera, 10 exemplars, 10 species per genus, shown in different colors here. And what we have done is we have done we have taken the distance matrix that we got out of continuous progressive distance, then done multidimensional scaling to do the best possible 3-dimensional projection of that distance matrix, and that's what I'm showing. And biologists were very happy because they did much better at clustering these 5 different genera than other methods had done. We thought it was awful. I mean, I'm showing you here the best view of the 3D diagram, and that was one thing that we felt it wasn't as good as it should be. The second thing was that when they saw that as an intermediate result in our analysis, we produced a mapping between the surfaces, they were really focused on those mappings. And they said, but you know, in this particular case, the mapping is not so good. And I looked at that case, and I looked at the distance. I said, but it's a very large distance. I don't trust that result. He said, this is one that you compute. I said, well, it's not as relevant. And that never really, it came out of the program. It wasn't a map. They didn't like it. For about, we had in these collaborations, I tried to have weekly meetings, sometimes it's 2 weeks because somebody's out of town, and for about a year, our conversation stalled. We revisited the same points over and over again. And that's when I finally realized, I said, you guys are actually no longer interested in the distances. You're interested in the mappings. And they had never said that, you say. They hadn't articulated that. I'm not even sure they had realized it. But once I articulated it, they said, yes, that's right. So I said, well, then we'll change. We'll look for mappings. And so that was one thing. The second thing, and that was that Ting Ran, I had just become my graduate student, and he said, but you have distances, and there is this method that can reparameterize data based on distances where you emphasize short distances. You use these diffusion methods. You use what I explained earlier to reparameterize. And so he said, let's reparameterize. Let's go to a larger dimensional Euclidean space, 50 dimensions. Let's take the distances there and then do an MDS. And that's what you got. And that made a big difference. First of all, the clustering was much better. So it showed that the whole approach, this reparameterization, made sense. It was doing something. We always try to link it back to things that are visual and that biologists can inspect. And that have to make biological sense, either because they tell us or because here the clustering is much better based on biological criteria. So because we want to use the domain knowledge without having it nicely encoded in mathematical criteria. Okay, so that's when they first started believing that, yes, small distances are more trustworthy, and we can encode that. Because all the pairs for which the mappings were not good, were things in which distances were much larger. But in fact, and that was then Ting Ran's thesis, and that's what I want to tell you about in the last part of this talk, he defined another way of dealing with the data, the same data, which did much better, which he calls the horizontal diffusion distance, horizontal-based diffusion distance. Okay, let me tell you about that. To get the diffusion distance, what we did was we used the local distances on the collection of teeth. So every tooth is just one point. And we have defined distances between points. And we knitted together neighborhoods, things that were close, in order to do a spectral parameterization of the collection. And that gave us a distance. So we have all these teeth, and we defined our mappings in the process of defining distances. Then we had the distances, and we have representation. But in that process, we have those mappings. We can use the mappings. So far, I've used them only to obtain numerical values for the local distances, for these local diffusion distances. You can do much more. You can, in fact, view what you have as, this is a fancy word for something, as a fiber bundle. I mean, mathematicians like to think of cases where you have something that has parameters that vary, but where to each point you associate something that has structure itself. Some of these cases you think of as a fiber bundle. So what you have is a manifold, and to each point you associate another manifold. So think of it as skin with hair. I mean, then if you have a way of going from one fiber to the other, so mapping hairs to each other, you call that connection. In our case, the connection we look at is actually a very, for mathematicians, a very trivial one. Because we believe that if you compare tooth A to tooth B, or A to C and B to C, but if they have a common ancestor, D, then you could do the right mapping from A to B and B to C and A to C by first going to the ancestor and then come down again. What that means is that the whole thing is going to be commutative. When you go from A to B or from A to C and then from C to B, and that means it's a really boring connection by mathematician standards. It has no holonomy, and they say, why would you? But we do anyway. So what you can do is look at the whole thing, the thing in which each point has two labels, namely on what of these teeth am I sitting, and then where on the tooth I'm sitting. So these two labels. So you take everything with these two labels and you'll get an enormous collection, and you can diffuse on that. You can diffuse on that because you know how to hop from one tooth to the next. You have a mapping. And you know you can attach a probability to that because you have an approximate distance. For the moment, let's believe the continuous progress. In the beginning, we were so proud of it. And CP, continuous progressive, and now we call it the crappy map. So it's the first map with which you start. And so you have a way with which you can build a diffusion from tooth to tooth, and you can diffuse on each tooth. So you can build a diffusion on the whole thing. You can, and so here's where the fiber bundles and so on. What you can do is you can, so let's get here. I can look then at, with that diffusion, to the diffusion on the whole manifold and look at the eigenvectors, like I did before. The eigenvectors will have, of course, evaluations in every of those points. So some of them on the same tooth, some on different teeth, and I've shown that with different colors here. Note, for this point, I do not even need a similar number of points on each tooth. It is not necessary for this approach. So that was something that was a big plus for the biologists. I take care of it in my mapping. I have to then do some sophisticated things to say that the mapping is not everywhere defined and so on, but you can take that into account. If my mappings were perfect, then in fact what would happen is that the eigenvectors, I mean, these are orthogonal eigenvectors, if the geometry was perfectly transported, if the teeth had been identical, let's say it that way. If I had identical teeth, then I would expect the restriction of the eigenvectors to the first tooth and the second tooth to be exactly the same. And so I would imagine that the gram matrix of the restrictions to one tooth would be exactly the same as the gram matrix of the restrictions to the other tooth. There. So I can measure how similar teeth are to each other by looking at the difference between those gram matrices. So that's a way of reducing distance to a much smaller dimension. I keep L eigenvectors, I make their gram matrix, and a reason the biologists immediately liked this and immediately ran with it, I said, don't, they said we want to, is that if you then looked, as I told you, I always want to see what things amount to. So I said, let's, in this huge dimensional space, let's imagine we try to cluster. Try to find points that belong together. Imagine that you make 13 clusters. These are colored in different colors, the clusters you get. So you have, for the yellow clusters, you have points on every tooth that in this large dimensional space, this reparametrization, are closer to each other than to any of the blue points, for instance. First thing to notice, and this was not a given, is that every color is represented on each tooth. The second thing to notice is that even though we started with correspondence maps that were area preserving, we have not preserved areas in this way of looking at it. So the area preserving was there to make a noisy, a crappy version, and the whole analysis has tried to denoise that and has gotten rid of the area preserving because it doesn't make biological sense. So the biologists were really happy with that. But not only that, they really liked that the regions that were demanded. I mean, they know about evolution and growth of teeth and so on, and they felt that these made a lot of sense to them. And so they immediately started mapping similar regions that are colored similarly and taking that correspondence map to do things and test other hypotheses with. Okay, but so this is what I told you. We take these restrictions and we take the gram matrices, and you can then look as a possible distance between two surfaces as the distance between their two little gram matrices. I mean, weighted with eigenvector. And this is how we got this. And when the biologists saw this, oh my God, they say some groups are closer to each other than to others in this view where you isolate them best. Maybe it shows evolution. I said, come on, I mean, we're looking at just one tooth. I said, well, Cuvier did it. I said, come on. And indeed, it did not show evolution, but it did show these are the species. And so you see purple and blue are together, and there are in this phylogenetic tree actually the furthest away from each other, but they eat the same thing. We're looking at molars. So it really clustered together nicely by what they were eating. And so it had biological signal. So we were really onto something. But you can do something else. You can also say, well, for every tooth, if I take the 3 dominant eigenvectors, then I can have a 3-dimensional embedding. A 3-dimensional embedding of all these teeth, it looks like a little Pringle. And that's really because of the eigenvectors of the Laplacian. I mean, you have one that shows plus minus, and another one that shows plus minus, and then a third one that does plus plus minus minus, and you get something like it. So that you get a Pringle is not so surprising. But that they look very similar, that they look all like the same Pringle, is very nice. So what we really have in a sense is here a nice parameterization of the template, of the thing, of the fiber, the abstract fiber. And this allows us to register, because we took our 500 points on one tooth and our 500 points on the other tooth arbitrarily. We didn't use landmarks, so of course they were not going to correspond exactly. But what we can now say is, look, this one blue point here lies in between those red points with these barycentric weights. So the corresponding blue point there should really be mapped to the corresponding barycentric weight thing of the red points. And we have a much better parameterization. And in fact, that does work. I mean, here we have an example of points. So what I did is, let's try it and let's track it. And so I asked a student who did this to look at points and look at the ones that moved most. And I asked biologists, look, this is where we move the point to based on this new registration. Does this make sense for you? And I said, oh yes, this is the right direction. So we like that a lot. So I'm getting near the end here. Other things we have been doing is using the register maps to do Gaussian landmarking. That's to say, instead of, so now we have our understanding of these teeth and so on, we can now say we have them all as functions on a template. Now we can say, where would we like to landmark them if we use kind of Kriging process, a Gaussian landmarking. We say we want to find the point from which the sample of which we learn the most. And then the next point, given what we've learned from the first point, where we learn the most, and so on. And if you do that, you then end up with a landmarking that is something that the biologists very much like as a landmarking process. And I'm here to show the difference between if you try to do a local maximum of just Gaussian, of just curvature, or geodesic farthest point sampling, are much less interesting biologically than the Gaussian process landmarking. Finally, what do I have here? Yes, this is actually, the first one I was doing here is Gaussian landmarking for one tooth, but based on diffusion for a tooth. Here is the Gaussian landmarking basing on the whole collection, which is what they really are happy with. And I think that's probably, and here's another one, and that's probably or the last thing I have to show. And that last picture was actually, I found in my email this morning. It was just from a student who sent it to me. I'm giving a talk about this. Get me a picture. So, other generalizations in progress. Well, we want to look at prosthesis distance that is locally geometry adapted, so where we take curvature into account to define that local Kraepelin mapping that we then going to denoise. And we have a much better way of doing a DNE with an algorithm that is much more robust implementation. What happened there was, so Jaron Lippmann had this beautiful package that did DNE. And they used it, he had incorporated the parameter limitations that they gave him. What's the range over which we'll use them and so on. And so they were so happy with it that over the years they started using it, stretching those boundaries. At some point it started breaking. But they didn't tell us. I mean, it was such a magical thing. They developed kind of rituals for using it. If you first do this and then it doesn't break as much. And when I finally learned, I said, but you should never, ever, ever do this. What we're doing is doing a numerical implementation of a mathematical concept. And the numerical implementation has limits, but the mathematical concept should be the one. And so if you go outside those limits, then we'll implement it differently, but so that we still do a valid approximation of that mathematical concept. And so I said, don't invent a ritual that may help. And then Shanshan implemented Ariadne, which works for many things, other biological things than just, and which we are now. We want to use where that field, that the NE field, scalar field is maximal in the. And then finally, I kind of swept this under the rug, but when I said we build a diffusion on the collection, we had a diffusion between teeth, and we have a diffusion on each tooth. And because it's locally a tensor product, you just make the product of the matrices and you diffuse. Well, what you usually do is you do a Trotter product formulation, a Trotter product of the small time diffusions in order to get the diffusion over the full thing. But you don't know how to match the diffusion time on the teeth with the diffusion time between teeth. So that ratio is something for which we don't really know how to do that. But our interpretation is that we first want to set it in such a way that you diffuse over the collection so that the dominant eigenvectors will be given by the smaller scale diffusion on the tooth. We want to learn the template. Learning the template will teach us the correspondence maps. Once we've learned the correspondence maps, we can change that ratio in order that the dominant eigenvectors will be given by the diffusion over the base manifold, and we can use that parameterization to learn more there. But we're not there yet. I'm going to stop here.
Any questions for Ingrid?
Yes.
If you have like maybe some rare fossil teeth that have like damage, so you have many of them, small damaged regions, different damaged region on every tooth. Is there a way to make the method ignore the damaged regions so that you can still get a good, like, smooth mapping?
So that has to do with the fact that you have to want to be able to deal with missing regions.
Yes.
So, and yes, our method can be adapted to that. So you do diffusion in places where you have that. So for the moment we we ask them to mark it. Because it's only when they're very special teeth that they want to include. Otherwise, for the others, they have so many that they can say, okay, this one, let's toss it. I mean, they toss so many. So, but if it's a very special one, then they're willing to kind of mark it, and we can do it.
Very cool.
Can you ever like impute missing data using something like this too? Like, if you have
Okay,
a
so what we can is try other manifold methods in order manifold learning. I mean, local manifold learning. So we don't do missing data on the base manifold, but on each tooth we can try to build in the best possible interpolation, and we can taking into account neighbors. We are working on a method of doing that as well. So I have to drink something. We also have developed, because we want to, so we have all these theories and we test them with the domain knowledge of the biologist. But we'd also like to test them just mathematically. So we have now developed a generative model to build something that looks to biologists as teeth. And it took a while before we were there, because and so where we can control the parameters, and we want to investigate how well do we learn the base manifold, which we now know in that case. I can't tell you more about that. Yes.
So I'm interested, when you want to model things that are pointy, you want to move out of the manifold setting.
Yeah.
Would you ever want to use an orbifold, or what other kinds of structures?
I expect that you would use that. You would then have to have a good way of defining diffusion among those. And I don't know how to do that, but ask Jonathan. He has ideas.
Who will?
Yeah. Yes.
Do you have like to hear insights on the generalizability of these two, perhaps, you know, data that are more noisy, that are
Yes.
not, for example, I'm interested about biological data, gene expressions, which are very high dimensional, and you assume that there is some low dimensional structure on it, but they are usually measured with much higher noise level. And do you have insights on like this kind of approach at what point? In terms of the signal to noise ratio, like how precisely you measure them?
So I don't have a direct answer for you. What I do know is that in cryo-EM, where you have extremely high noise on every one of your images, Amit Singer has been using diffusion methods for many years in order to get much, much sharper images out of the cryo-EM data. So parameterizing by means of these diffusion methods really can be noticed significantly in that case.
Great. Please join me in thanking Ingrid again.